The overall goal of the following experiment is to use microbead implantation to introduce alterations in the local tissue environment at specific spatial and temporal points during zebrafish embryogenesis. This is achieved by first preparing a microbead implantation tray to facilitate the precise handling and implantation of the microbeads. As a second step, the zebrafish eggs are incubated to the desired stage of development. The microbeads are then prepared in the appropriate experimental solutions and carefully transferred into the desired location within the embryo. Ultimately, microbead implantation can be performed without the disruption of normal zebrafish development, allowing assessment of the effects of the molecules of interest on zebrafish embryogenesis.
This method can help answer key questions in the developmental biology field, such as how do different molecules affect the process of organogenesis?
Generally, individuals new to this method will struggle, as it takes practice to become skilled at handling the microbeads.
To prepare the microbead implantation tray, first heat freshly prepared 2% agarose solution for 90 seconds in a 250 ml Erlenmeyer flask, swirling the flask every 30 seconds to prevent the gel from bubbling over. Next, place the lid of a 60 by 15 mm Petri dish into a 150 by 15 mm dish with the interior of the lid facing upward. Then, anchoring the lid of the smaller Petri dish with an index finger, pour the hot agarose gel into the bigger dish. Allow a thin layer of agarose to form below the smaller lid to prevent the formation of condensation and to make the beads easier to see under the microscope. As the agarose cools but before it solidifies, slowly angle a well mold into the top of the gel to allow easier placement of the embryos.